Welcome to today's deep dive. If you've ever looked at a medical scan or read about some new genetic discovery, you've probably wondered how scientists actually see what's happening inside us.
Right. Yeah, like what are they actually looking at?
Exactly. Well, today, our mission is to explore a breakthrough that fundamentally changes that view. We are diving into a newly published resource from the Nucleic Acids Research Journal called So3D, which stands for Three-Dimensional Spatial Omics.
It really is a massive leap forward because, to put it in perspective, for decades, analyzing human tissue has been like looking at a flat 2D paper map of a massive sprawling city.
A map where you can't see the buildings.
Right, exactly. You can see where the buildings are, technically, and you can map out the roads, but you are completely locked out of the buildings themselves. You have absolutely no idea what the people inside are actually doing.
Which is so frustrating from a research perspective. And today, we're talking about upgrading that flat paper map to like a fully interactive 3D Google Earth model.
Yes, a perfect analogy.
A model where you can virtually fly through the tissue, peel back the roof of every single cell, see exactly who lives there, what their specific job is, and this is the part that completely blew my mind in the research, actually intercept the chemical text messages they are sending to the cell next door.
That interactive capability is the core of S3D. What the researchers have done here is curate over 1.1 million biological spots across multiple species to build actual navigable 3D architectures of tissues.
Over 1 million spots. That's just wild.
It's a huge undertaking. And today we're going to walk you through how this next generation tool was built and how it's, completely rewriting our understanding of everything from the chemical cloaking devices used by breast cancer to the way a fruit fly embryo physically wires its own nervous system.
Let's start with the sheer scale of the construction project here, because before we can use the 3D map, we really need to understand how hard it is to build one.
Oh, it's incredibly difficult.
Right, because for a long time, the gold standard in biology was single-cell RNA sequencing, which is great, right? It tells you exactly what genes a cell is turning on or off.
Exactly. You get the full genetic profile.
But to do that, you essentially have to throw the tissue into a blender. You get all the genetic information, but you completely lose the address of where that cell originally lived.
Yeah, you're left with this genetic soup. And then we eventually moved to spatial transcriptomics, which was a step up because it allowed us to keep the address, but only on a microscopically thin, flat 2D slice of tissue.
And the limitation there is pretty obvious.
Right. Life doesn't happen in a flat plane. A tumor growing, a heart beating, a brain processing a thought. These are highly coordinated events happening in three-dimensional space. The So3D database was built specifically to solve that spatial blindness.
And the volume of data they used to solve it is staggering. We're talking about 72 different data sets spanning 4 distinct species: human, mouse, fruit fly, and the axolotl.
The axolotl inclusion is particularly fascinating, I think.
Oh, because it's a salamander that can regenerate.
Exactly. It can completely regenerate severed limbs or damaged spinal cords. So when you combine those 4 species, the database covers 15 different tissue types and 5 major diseases.
Wow.
Yeah, they integrated 882 individual physical tissue slices, which gives us that 1.1 million spatial spots, and then they anchored all of that with single-cell RNA sequencing data covering over 760,000 individual cells.
Okay, but this brings up a massive logistical question for me.
Yeah.
If I have, like, a loaf of raisin bread, and I want to understand its 3D internal structure, cutting it into 100 paper-thin slices and then trying to perfectly align photos of those slices back together digitally, that sounds impossible.
It sounds impossible because it essentially was for a very long time.
Right, because if the blade shifts even a fraction of a millimeter or the tissue warps, aren't the cells going to be wildly misaligned when you stack them in a computer?
That exact alignment problem held the entire field of spatial biology back for years. You really can't just stack the images. To overcome this, the So3D platform relies on a sophisticated algorithmic framework called Stitch 3D.
Stitch 3D, right.
Yeah, and it essentially uses deep neural networks to perform what we call spot registration. And it leans on 2 very specific mathematical algorithms to do it: the iterative closest point, or ICP algorithm, and the paste-to algorithm.
Okay, let's unpack those. I can guess what paste-to means in a general sense, but what are these algorithms actually doing to the tissue slices? Let's start with ICP.
Sure, so think of the ICP algorithm like trying to align two transparent maps of the night sky that were taken from slightly different angles.
Okay, I'm picturing that.
The algorithm searches for overlapping constellations. In this case, these are distinct patterns of cellular landmarks that exist on both slices.
So it's looking for recognizable shapes.
Exactly. Once it finds those shared constellations, it mathematically rotates, tilts, and shifts the digital slices until those specific landmarks lock together in 3D space.
So ICP handles the rigid rotation. What about paste?
PASTE is a bit more elegant, actually. It stands for Probabilistic Alignment of ST Experiments. It uses a concept from mathematics called optimal transport.
Optimal transport. What
Yeah.
does that mean in this context?
Well, imagine you have a pile of dirt and a hole you need to fill nearby, and you want to move the dirt with the absolute minimum amount of effort. PASTE looks at the genetic profiles of the cells on slice 1 and the cells on slice 2 and calculates the most biologically probable way they connect across the z-axis.
So it's ensuring that, like, a blood vessel on one slice smoothly continues into the blood vessel on the next slice, rather than jaggedly jumping to the side.
Perfectly said. Yes, it maps the most efficient, logical, biological path between the layers.
That makes perfect sense. But what about the physical environment? I mean, say a lab technician slices half the tissue on a Monday, goes home and slices the rest on a Tuesday. The room temperature might be different. The chemical stains might sit for 2 extra minutes. Doesn't that change how the cells look to the computer?
It absolutely does, and that introduces what we call batch effects. Even if two cells are functionally identical, the Tuesday cell might look genetically different to the machine simply because of environmental noise.
Just because it was a different day.
Right. And this is where the deep neural networks inside Stitch 3D really shine. The system pulls in that blended up single-cell RNA data we mentioned earlier. It uses that pure data as a baseline truth to mathematically scrub away the environmental noise. It basically irons out the batch effects.
Okay, so let's do a quick recap here and make sure we're on the same page.
Good idea.
We take the physical slices, we use optimal transport and constellation mapping to perfectly stack them, and then we use neural networks to erase all the human error and lab noise. And the end result is a pristine, unified 3D digital block of biological truth.
That's a great way to put it. You now have a perfect 3D replica. But having the map isn't the same as understanding the territory.
Right. Staring at a million raw data points doesn't give a scientist any actionable insight. You need a dashboard to navigate it.
Which is exactly what So3D provides. Imagine you're holding this interactive 3D hologram of a human organ. How do you start exploring it? The researchers built a 5-tool Swiss Army knife to do exactly that. And the way these tools flow into each other is basically a masterclass in biological detective work.
Let's walk through those 5 tools.
The workflow is highly intuitive. It starts with the first module called Domain 3D. When you look at a massive block of tissue, you first need to understand its macrostructure.
The big picture.
Right. Domain 3D scans the 3D space and groups together spatial domains that share coherent, similar gene expression.
So going back to our city map analogy, Domain 3D is drawing the zoning borders. It's looking at the activity and saying, okay, this area over here is clearly the industrial manufacturing district, this quiet area is a residential suburb, and this highly active zone is the financial center.
Exactly that. It gives you the biological neighborhoods. But knowing the neighborhood doesn't tell you who actually lives in the houses. For that, researchers moved to the second tool, which is Cell Type 3D.
Cell Type 3D. Got it.
This module pinpoints the exact physical coordinates of different cell types. And it has to perform a highly complex mathematical task called deconvolution to do it.
Let's unpack deconvolution, because a single spot on these spatial maps isn't always just one neatly packaged cell, right?
Far from it, unfortunately. Depending on the technology used, a single spatial spot might encompass 3, 4, or even 5 different cells all squeezed together. It's basically a biological smoothie.
A biological smoothie, that's a great visual.
Right. So Cell Type 3D looks the mixed genetic signature of that smoothie, compares it against the pure single-cell reference data, and mathematically untangles it. It can tell a researcher, hey, this specific microscopic coordinate is exactly 60% macrophage, 30% T cell, and 10% structural fibroblast.
That is wild. So we've zoned the city, and we've identified the exact professions of the people living in every apartment block.
Yes.
But a cell's identity doesn't tell you what it's currently doing. That brings us to the third tool, right? Spatial DEG 3D. And DEG stands for differentially expressed genes.
Exactly. And this tool highlights why the 3D aspect is so crucial. Spatial DEG 3D identifies region-specific biomarkers. It looks at how the exact same type of cell behaves differently depending on its physical location.
Right, because location dictates stress. Like, a tumor cell sitting on the outer edge of a mass has access to plenty of oxygen and blood, but an identical tumor cell trapped dead in the necrotic center of the mass is suffocating.
Exactly.
So spatial DEG 3D lets you see how that suffocating cell is flying completely different genetic flags at its window just to survive.
It perfectly maps that spatial heterogeneity. But, a flag is just a genetic marker. To a doctor, knowing gene X is turned on isn't fully actionable. You need the fourth tool, which is Function 3D.
Function 3D.
Yeah, this module takes those active genes and analyzes the biological pathways they belong to.
I noticed in the sources they use databases like the Gene Ontology and KIGG. How does the Function 3D actually use those?
Think of those databases as massive biological translation dictionaries. Function 3D takes the active genes identified in a specific neighborhood, runs them through the dictionary, and translates the raw code into a biological action.
Oh, I see.
Yeah, it turns genes A, B, and C are active into the actual insight. This specific 3D neighborhood is actively building new blood vessels.
So we have the neighborhoods mapped, the residents identified, their stress flags spotted, and their daily jobs translated. Let's do another quick summary check. We've gone from macro to micro.
We really have.
But the fifth tool is where the science fiction element really kicks in for me. Communication 3D. When I read how this worked, it sounded like researchers were literally wiretapping the cells. Like we aren't just seeing that cell A and cell B are neighbors, we are actually intercepting the chemical text messages they send to each other.
Wiretapping is a remarkably accurate description. Cells obviously don't have mouths or ears, right? Their entire language is chemical. One cell secretes a molecule called a ligand. Yeah, which you can picture as a highly specific chemical key being tossed into the physical space between cells, and then a neighboring cell has a receptor on its surface.
The keyhole.
Exactly, the keyhole. Communication 3D maps every single one of these ligand receptor pairs in 3D space using an algorithm called cell chat.
So we are moving from a static photograph of the tissue to a dynamic movie of who is coordinating with whom. You could literally trace a distress signal leaving a dying cell and watch it travel across the 3D space to bind with an immune cell 3 layers away.
And that capability completely changes how we approach disease. Which brings us to how these tools perform outside of a computer simulation. To prove the power of SO3D, the researchers deployed this exact 5-tool Swiss Army knife onto a real-world battlefield, human breast cancer.
Let's look at this case study because this really cements the concepts. They loaded a breast cancer or BRCA dataset into the Town 3D platform. We're looking at 1,729 spatial spots mapped across 3 consecutive slices of tissue. When they unleashed the first 2 tools, the zoning and resident identification, the algorithms immediately identified 5 distinct neighborhoods, which they labeled clusters 0 through 4.
And the contrast between these clusters was stark. Cluster 0 was flagged instantly as an immune-enriched zone. It was packed with B cells and macrophages, largely identified by a highly localized gene called IGH1.
So this is the body's defense force.
Yes, gathering and staging an organized front. In contrast, clusters 1, 2, and 4 were heavily fortified zones dominated almost entirely by invasive tumor cells.
It's literally a map of a microscopic war zone. You have the immune army amassed in cluster 0 and the tumor fortress occupying clusters 1, 2, and 4. But to understand the war, they had to look inside the fortress, right?
Exactly.
So they ran the 3rd and 4th tools, Spatial DG 3D and Function 3D, to see what the tumor was doing. And they found something highly unusual involving a gene called DHCR24.
This was a critical insight. Inside the tumor fortress, the expression of DHCR24 was massively upregulated. And when the Function 3D dictionary translated that gene, it revealed that DHCR24 is a master driver of cholesterol biosynthesis.
Wait, normally when I hear cholesterol, I think of, arterial plaque from eating too many cheeseburgers.
Yeah.
Why is a breast cancer tumor running a massive cholesterol factory in the middle of a war zone?
It's a great question. It's because cholesterol is an essential structural component of cell membranes. If a tumor intends to proliferate, if it's trying to rapidly divide, expand its borders, and eventually metastasize to other organs, it requires an immense supply of raw building materials to construct new cell walls.
Oh, wow.
Yeah, the tumor in clusters 1 and 4 was using DHCR24 as a localized fuel pump to drive its own physical expansion.
So we know that the tumor is building a wall, and we know the immune cells are amassed nearby. This is where the 3D wiretapping becomes crucial.
It really is.
Because if you just blended this tissue up in the traditional way, you just see a messy soup of immune genes, tumor genes, and cholesterol genes. You wouldn't know the layout. But because they use communication 3D, they could actually listen to the radio traffic on both sides of the border. What were they saying?
The wiretap revealed a highly sophisticated level of chemical warfare. First, they monitored the immune cells in cluster 0. They intercepted a communication channel relying on a ligand called RARES2 binding to a receptor called CMKLR1.
Okay, let's call the RARES2 CMKLR1 channel the flare gun pathway, just to keep it simple.
That's perfect. It operates exactly like one. The immune cells were constantly firing these RARES2 chemical flares into the surrounding 3D space, desperately trying to recruit more immune cells to the front line to attack the tumor mass.
But the tumor isn't just passively taking the hit, is it? When the system tapped the radio lines inside the tumor fortress, they found the cancer cells were using a completely different communication loop. The APP ligand binding to the TNFR-SF21 receptor. What does that specific text message say?
That pathway, the APP-TNFR-SF21 axis, is what we call an autocrine loop. The tumor cells were sending messages to themselves and to the other cancer cells immediately adjacent to them.
What's the message?
The message essentially commands the local environment to become immunosuppressive. The tumor is coordinating with its neighbors to project a chemical cloaking device, neutralizing the immune cells before they can cross the topological border of the tumor.
That is chilling. We aren't just guessing that tumors evade the immune system. We're looking at an interactive 3D map, isolating the exact XYZ coordinates of the cancer and reading the exact encrypted message it uses to turn itself invisible to the body's defenses.
And this highlights exactly why the 3D architecture is mandatory. In a 2D slice, you might see a few immune cells near a few tumor cells, but you miss the topological sphere the tumor forms to shield its necrotic core. In 3D, you can see the exact physical depth and boundary of that cloaking field.
It makes you realize how incredibly blunt traditional chemotherapy is. We blast the entire body with toxins, hoping to hit the tumor. But if we can map the exact local radio frequencies a tumor uses to hide, we're looking at a future where we just deploy a targeted drug that jams that specific APP-TNFR-SF21 signal.
Exactly.
Stripping away the cloaking device and letting the immune system do its job.
The therapeutic potential is immense. But, to truly appreciate the So3D platform, we have to look beyond disease. Understanding how systems break down is only half the battle. The other half is understanding the fundamental rules of how healthy life builds itself.
That's a great point.
And the researchers also turn these tools on human brains and fruit fly embryos.
Let's look at the brain application first. They mapped the human dorsolateral prefrontal cortex. That's the area of the brain responsible for our highest order cognitive functions. I would imagine a 3D map of the brain's wiring is incredibly dense.
It is, but So3D cut right through the noise to map the distinct layers of white and gray matter. The true revelation came when they looked at the communication networks maintaining that wiring, specifically focusing on oligodendrocytes.
Now, for those who might not recall from biology, oligodendrocytes are the specialized cells that wrap themselves around the long, wiry axons of your neurons. They provide insulation, like the rubber coating on a copper wire, so that electrical thoughts can travel at high speeds.
Right, exactly. And the assumption might be that they just quietly do their insulating job alone. But the 3D mapping proved otherwise. It showed oligodendrocytes actively and constantly communicating with astrocytes.
Astrocytes. Those are the star-shaped support cells that regulate the brain's environment, right?
Yes. They were talking via collagen signaling, specifically tossing a ligand called COL4A5 to SDC4 and ITGAV receptors.
So building and maintaining the brain's physical hardware isn't a solo operation. It's a continuously negotiated, dynamic conversation between entirely different families of cells across 3D space.
Precisely. And if you want to see an even more dramatic example of cells organizing in 3D space, we have to look at the Drosophila, the fruit fly dataset. They analyzed a developing fruit fly embryo, utilizing over 31,000 spatial spots across 21 slices.
I found this dataset the most captivating. Because you aren't just looking at static tissue, you are literally capturing the exact spatial coordinates of a living organism physically assembling itself from scratch.
The clarity of the maps is unprecedented. They could perfectly trace the emerging architecture of the central nervous system and the formation of the foregut, midgut, and hindgut.
They could actually watch the gut form.
Yeah. They tracked specific genes like a PULP and PLP2, which handle lipid transport, and watched exactly where they fired up in the midgut to manage the embryo's metabolism. And because they were finally looking in true 3D, they even discovered entirely novel tissue marker genes, like one labeled CG32073, that science had simply missed before.
But I have to play devil's advocate here for a second. Mapping a fruit fly embryo is a really cool science experiment. But why should we as humans care about the gut tube of a fly? Does analyzing a fly, or an axolotl for that matter, actually give us a cheat code for human biology?
It gives us the master blueprints, because evolution is incredibly conservative with its best ideas. The fundamental physical and chemical rules of biology, how a cluster of stem cells figures out what is top and what is bottom, how a tube forms, how cells know when to stop dividing, these architectural mechanisms are highly conserved across millions of years of evolution.
So by watching the fly assemble itself, we are essentially reading the rough draft of human embryogenesis.
Exactly. If we can use the S3D platform to reverse engineer the exact 3D chemical blueprint of how a fly builds a functional nervous system, or how an axolotl reads its spatial environment to perfectly regrow a severed spinal cord, well, we acquire the exact molecular vocabulary needed to trigger those same regenerative processes in human patients. You cannot repair a complex machine if you don't possess the original 3D engineering schematics.
And for the first time, we actually have the tools to read those schematics. It really feels like we are crossing a threshold here.
We are.
I mean, we started this deep dive talking about the frustration of flat 2D city maps. The era of just knowing what genes you have in your body is ending. What so 3D and spatial omics represent is the realization that the future of medicine is fundamentally tied to geography.
It's a profound shift in perspective. It's no longer just about the genetic code. It's about the zip code. Where are those genes located? What 3D neighborhood do they inhabit? And who are they conspiring with across the cellular borders?
It makes you wonder what happens when this technology scales even further. Imagine applying this level of precise 3D spatial mapping, not just to understand cancer, but to reverse engineer aging itself. What if, 10 years from now, you could take a true 3D spatial scan of a failing liver, identify the exact coordinates where the regenerative communication lines have broken down, and essentially use a biological 3D printer to patch the organ with perfect, locally coded replacement tissue? We wouldn't just be treating symptoms. We'd be actively reprogramming the spatial software of human biology, something to mull over the next time you think about how your own cells are organizing themselves right now. Until next time.